name is Kim Hobbs, and I live in Conception Bay South, Newfoundland and Labrador. I have a Master of Science degree in Biology from Memorial University, and I work as an aquatic science technician at Fisheries and Oceans Canada. My job is to provide field and laboratory technical support to research being conducted by the Aquaculture, Biotechnology, and Aquatic Animal Health section.
Laboratory work is conducted at the Northwest Atlantic Fisheries Centre in St. John's, Newfoundland. Samples taken in the field are prepared for chemical, biochemical, histological, and genomic analysis using various pieces of analytical equipment. Weights and measurements of fish and shellfish are taken for morphometric analysis, and tissue samples are taken for lipid, enzyme, histological, and genomic analysis. Tissues for histological analysis are placed in plastic perforated tissue cassettes and then placed in a machine called a tissue processor. Here they are dehydrated and infiltrated with wax. At the end of processing, the tissues are embedded in paraffin wax, which stabilizes them during sectioning. Thin sections of 7 micrometers or 0.007 millimeters thickness are cut from the wax block using a rotary microtome. These sections are then mounted on slides, stained, and viewed under a compound-like microscope to visualize cellular detail. To view the surface structure of a sample at high magnification, a scanning electron microscope is used. Samples are coated with an ultra-thin coating of an electrically conducting material before scanning. Samples can be magnified to more than 500,000 times, which is about 250 times the magnification of the best light microscopes. For genomic analyses, genetic material, either DNA or RNA, is extracted from tissue, purified, and amplified. To extract the genetic material from the cells are disrupted in a tissue lyser. The genetic material is then separated from other cellular components and purified for species identification, population genetic studies, or gene expression studies. Aquaculture research allows us to understand interactions between aquaculture and the aquatic environment, to increase our knowledge of pests and pathogens affecting cultured species, and to work with industry partners towards a sustainable and environmentally responsible industry. Fieldwork is conducted in and around finfish and shellfish aquaculture sites using a variety of specialized equipment for the collection of physical, chemical, and biological data. A CTD, or conductivity temperature depth sensor, determines physical properties of seawater, such as salinity and temperature at different depths. This CTD also has a chlorophyll A sensor. Chlorophyll A is the primary photosynthetic pigment in phytoplankton. By measuring chlorophyll A, we can estimate the amount of phytoplankton in the water. Niskin bottles are used to take water samples at a desired depth. To collect a sample, the stoppers at both ends of the bottle, which are connected on the inside of the bottle by elastic cords, are held open by plastic cords attached to a release mechanism. The bottle is attached to a cable and lowered into the water. When the desired depth is reached, a small weight called a messenger is released down the line, striking the release mechanism, which causes the stoppers to close the bottle, trapping the water inside. The water samples can then be used for a variety of analyses, such as nutrient content or to study plankton. Plankton nets are used to collect planktonic organisms of nearly any size, depending on the mesh size of the net. The mesh size is large enough to allow water to pass through, but small enough to trap the target organisms. At the end of the funnel-shaped net is a collection cylinder called a cod end. Plant, phytoplankton, and animal, zooplankton, samples are collected from the cod end into sample containers for analysis. To compare samples quantitatively, a flow meter is attached to the net to determine how much water passes through the net, allowing for measurement of abundance and biomass.
I hope you've enjoyed seeing some of the science we do at DFO. DFO scientists do everything from mapping the ocean floor and studying ocean conditions and climate change to stock assessments of commercially important species and monitoring pathogens and aquatic invasive species. If you're interested in a career at DFO Science, or you want to see some of the really cool science we do, visit our Facebook page. Check us out on the web at www.dfo-npo.gc.ca or follow us on Twitter at FishOceansCan. Thanks for watching.